Hello everyone. So in this session uh, we will discuss about the genes and chromosome structure, and we will also see how the DNA metabolism happens. So that is basically the replication of DNA. How the DNA replication happens. So before that, we need to understand what is gene and chromosome, right? So in the eukaryote, as you know, this is an uh, you know the DNA is organized in a form which is called chromosome, and we have multiple chromosomes for that. And uh, our chromosome or the DNA that is combined with uh, some histone protein in right in the nucleus, and histone and other histone like proteins, right, so that the a very large length of the DNA that can be adjust into a nucleus. So that's why DNA structure is important. DNA is folded. This DNA along with the chromatin protein, both of them, they will make a organization of right of this chromosomal structure, so that they can be adjusted within a nucleus. Right. So this is a bacteriophage T2 protein coat surrounded by its single linear molecular DNA. So this is just there is no chromosome. This is just example of a bacteriophage, right? So here this white one is the T2 protein that is surrounded by the single. This is the single linear molecule of DNA, right? So this is the most simplest one, right? But in the chromosome, the DNA is organized in a form of structure along with other proteins, right? So that it can be adjusted in the inside the nucleus. Now, what is gene? So, in the chromosome, which is the DNA only. So, the genes are the segment of a DNA, right? The segment of a DNA which can code for polypeptide chain and functional RNAs, right? So, meaning that the gene, for example, for us, the sequence which or the ORF which can actually code something that is called exon, right? And the sequences which cannot code anything, code which cannot code any amino acid, that is called intron, right? So gene can contain both exon and intron. So after the gene expression, when mRNA will be produced, then the mature mRNA will lack of the introns. It will have only the exons, right? So genes are the segment of DNA which can code for a polypeptide chain and the RNA. Right? Some genes are there which can code the RNA, right? Which cannot be translated to protein, but those RNAs are functional. For example, ribosomal DNA or rDNA region, they can code the ribosomal RNA, right? Which is not a protein, but that ribosomal RNA has a role to play, right? For the assembly of the ribosome or ribosomal biogenesis, right? So this is the gene, right? And then if you see from bacteriophage, if you go to the bacteria E. coli, then you can see the DNA from a lysed E. coli cell. This is the DNA electron micrograph of a uh, you know of a small circular plasmid DNA. So E. coli has plasmid DNA as well as the linear DNA, right? This is the linear form of the DNA in the E. coli, right? Which is the length is almost 2 micromolar, and this is also structured with some proteins, which is histone-like proteins in the E. coli, not histone but histone-like proteins. And also, apart from this, so these are the linear DNA, right? In the lysed E. coli cell, and these white arrows which are indicating these white dots, right, which are indicated by the white arrows, so those white dots are the circular plasmid DNA, right? This is the electron micrograph to give you idea how the linear DNA and the circular plasmid DNA looks like in a electron micrograph, which is obtained from a lysed E. coli cell, right? So till now we saw that there is linear DNA and plasmid in the case of E. coli. For bacteriophage we saw there is only linear DNA, which right. So now we will see what is the situation for the eukaryotes. For eukaryotes, DNA is distributed in a forms of different chromosomes, right? Here. So the entire DNA is this is your entire yeah. So this is your entire linear DNA, right? So that is uh, right. So that resides in a form of multiple numbers of chromosomes here, right? So basically in the uh, G2M phase, this chromosomes becomes visible, right? Because they becomes condensed, right? So that's why they can take a shape like this and they will be visible, right? So in the GTM phase specifically, when this linear DNA will be condensed, then it will take the shape of different visible chromosomes. So this linear DNA is divided into different chromosomes, right? And this is a electron micrograph of a eukaryotic chromosome, and which is actually taken from a human chromosome, right? So here you can see there are this is the centromere, and these are the telomeres, and there and after replication, one from this parental DNA strand, right? It has obtained the sister chromatine, right? So this is the eukaryotic chromosome electron micrograph structure, and so in eukaryote, apart from nucleus, also some organelles are there which can contain DNA, right? So, for example, mitochondria. There is a mitochondria have the mitochondrial DNA, so that can code some mitochondrial protein and mitochondrial RNA, right? So, which are required for the mitochondrial enzymatic activities and for the structure of the mitochondria, right? So that is for us, and yeah, and for plant also they have a extra organelle which is plastid, right? Plant and some other bacteria and fungi, especially the cyanobacteria, they have also the plastid, right? So this plastid also contains the plastidial DNA, right? So that will also express the RNA and the proteins which are required for the activity in the plastic. Right, so here is a chart of different organisms which is giving their DNA size and number of chromosomes. So for E. coli, there is only one chromosome, right, and which has a linear DNA, and number of genes are 4.405. For Saccharomyces cerevisiae, there are 16 numbers of chromosome, right, and this is the total DNA size, right, which is of course higher than the bacteria. For nematode, for example, C. elegans, they have 12 chromosomes, right? But they have the higher, number of size of the total DNA, right? In the plant section, Arabidopsis thaliana has 10 chromosomes. Fruit fly has 18 chromosomes. Oryza sativa, rice plant, it has 24 chromosomes. If we come to the mammals, then mus musculus, which is mouse, that has 40 chromosomes, and human, we have 46 number of chromosomes, right? So eukaryotic genes and chromosome structure are very complex, right? So because as I told you that eukaryotic genes can contain exons as well as introns. So in the transcription, when we will study the transcription, then we will see that after the transcription of the premature mRNA, the introns will be excised, right, to form the mature mRNA, which is called the intron splicing step, right? So that's why the gene can contain exon as well as intron in the higher eukaryote specifically. So this is example of a overlapping gene, right? This red portions are the exons, right? And these gray portions are the intron, right? So similarly here, hemoglobin beta subunit, it has three intron. One is 90 base pair, second intron is 222 base pair, and the third intron is 136 base pair, and it has two exons. Sorry, it has two introns. One is 131 base pair and 851 base pair, and it has three exons. First one is 90 base pair, second one is 222 base pair, and third one is 126 base pair. Right. So now, how this much length of the DNA can be accumulated inside the nucleus? The answer is by folding, by supercoiling, right? So that we will study now. So, in order to address this long length of DNA inside the nucleus, uh, right? So, for that DNA has to be folded and it should have a structural form, and some several proteins will come to help it. That we will see later. So, the DNA will take a form which is called supercoiling, means coiling over coiling, right? Because we know that DNA has a double stranded helical structure. So over there DNA can also coil, right? So that it will take less space. So you can accommodate a big chunk of DNA in a very less space, right? That is called DNA supercoiling, means coiling over coiling. So if you compare it with this uh, with this telephone wire, so so this, if you see this telephone wire very properly, then this portion is called coiling, right? Which is the case for the DNA, for a linear DNA, which is not supercoiled. There you can see that, like that DNA, it can also have the helical turn, right? So each helical turn means each coil. So that's why it is coiled. Now, if there is a coiling over coiling, suppose for example this part, right? See, there is a coiling over coiling. This is called supercoiling, right? Again, there are coiling over coiling. So this is called supercoiling. That means when this, right? So that means one strand of the DNA will cross the other strand. For example, here, here, and here, the second strand of the DNA is crossing the first strand, right? So that is called supercoiling. Here also, that is called supercoiling. For example, this is the DNA double helix which is coiled, right? When there is no supercoil, it is coiled, right? And now when it, right? So now when it will be supercoiled, then the second strand will pass over the first strand like this, and like this. So that will make a coiling over coiling, which is called as DNA supercoiling. Okay. So here is example where supercoiling is induced by separating the strands of a helical structure. For example, this is a double-stranded helical structure, right? Now there is no supercoiling, but when you take one of the strand, right, and you will pass the second strand over the first strand, right, like this, then you will tie a knot actually, right, which will create a supercoiling here, right, and right. So if you lose, suppose for example this is a rope which has two, uh, which is like a DNA, which has the two double-stranded two strands which are coiled over each other. That is called coiling. Now, if you hold this and if you just separate these two strands, right, like this, if you just increase the distances between these two strands, then in the middle portion, to accommodate this chain, in the middle portion there will be the supercoiling. It means one strand will coil over another strand, right, to maintain this, balance out this strain. That is called supercoiling, that can be induced by separating the strands of a helical structure. If you compare it with a DNA, if you separate two strands of the DNA, then it will induce the supercoil in the middle region, right? And this property, this supercoiling property, is a topological property, right? So what is topological property? Topological properties are the properties of an object that will not change under continuous deformation, right? For example, here, uh, as upon separating these two strands, this supercoiling has been induced, right? So now, the more you will do this, the more supercoiling will be there. So there will be no change, right? So the shape of the object that will not change under continuous deformation. For the DNA, continuous deformation include conformation changes due to thermal motion or an interaction with some protein, right? Discontinuous deformation means DNA strand breakage. When there is a strand breakage, then there will be discontinuous deformation, right? In that case only, these topological properties will change, right? Otherwise, if the DNA strand is intact, right, then it is example of a continuous deformation when the DNA strand is uh, intact, and this deformation is caused by interacting with some proteins or some motion, right? But the DNA strand is intact, so that is continuous deformation. So in this case, this topological property will not change. But when you induce a breakage here, then this is a discontinuous deformation. On that case, the topological properties will only change, right? So here you can see that as we studied about different forms of DNA, right? So here we will see the relaxed and supercoiled form of a plasmid DNA. So this is the relaxed form. There is no C, there is no supercoiling. But when one strand will go, yeah. So but when the one strand will pass the second strand for multiple times here, then it will create a supercoiling, that is right. So that's why it is supercoiled plasmid DNA. Right, now the degree of supercoiling will vary. Here the degree is the lowest, right? After that there will be more supercoiling, right? More passing of one strand over other strand. So this is, so here degree of supercoiling has been increased. Here it has been more increased, and in this case, in the last diagram, the this is the maximum supercoiling it can attain. Right. So from left to right, degree of supercoiling has been increased, right, for the plasmid DNA, and this is an example of a plasmid DNA which is circular and which is relaxed. There is no supercoiling, right? I think you can see the difference. See, there is no passing over of one strand over another, but in this case, there is passing over of one strand over the second strand for multiple times. So that's why it is supercoiled DNA, right? So. If you see that, so if you see what is actually the underwinding and overwinding, right, to explain the supercoiling property, right? So then we will see the effects of DNA underwinding, right? So suppose this is the initial form of the DNA which is relaxed, which has 8 turns, right, which has 8 turns. 1, 2, 3, 4, 5, 6, 7, 8, right? 8 helical turns are there, right? So now it is in a relaxed position. But when there will be a strain, suppose you were releasing 1 turn, right? So the turn will be reduced to 7. Then that is called the underwinding. When the linking number or the number of turns will be reduced, then it is not relaxed, it is strained, and this is called underwinding, right? So now this portion will, so because of this strain here, because one number of turn has been reduced, so because of this strain, to adjust this strain, the remaining portion will supercoil, right? So see, the second strand has passed first strand for one time. So here, right, so that is called supercoil. So now this strain is accommodated by formation of a supercoil, this strain of reduction of one time, right? So now this DNA DNA underwinding also makes the separation of strands more easier. For example, here you can see that each strand on underwinding should facilitate strand separation over about 10 base pair. However, the hydrogen bond base pair would generally preclude the strand separation over such a short distance, right? So now we will see that we will introduce a new term which is linking number, right? So that is used for the to understand the degree of supercoiling. So here, see, suppose one strand is actually, so if the one strand is passing over the second strand one time, then the linking number is 1, right? But if it is doing that by 6 times, then it is, the linking number is 6, right? So the definition is the number of times, uh, yeah, so the number of times the first strand will pass over the second strand, that is called linking number, which is 1 here and which is 6 here. Right? Now this DNA unbinding is or this you know linking number which we introduce, that is a topological property, because it does not vary when the DNA is bent or deformed, right? As long as the both DNA strands remain intact, right? So in this case, the linking number 6, right, or here the linking number 1, this will not change. Yeah. So this will not change until and unless the DNA is broken. When it will be broken, then the linking number will be changed. So if it is intact, and then if it will be bent and deformed, right, which is called as continuous deformation. If there is continuous deformations, right, then also this linking number will not be changed, right? So that's why it is called a topological property. But if you introduce the breakage here, then the linking number will be changed, right? So because there will be discontinuous deformation, right? So there is a term which is superhelical density, which is actually the degree of supercoiling, right? That measures the number of turns removed related to the number of turns present in the relaxed DNA, right? So delta Lk is the number of turns removed due to underwinding, right? That's why delta, which is the final linking number minus initial linking number, that is delta Lk, right, by Lk naught, which is the initial linking number of a relaxed DNA, right? So that is called the superhelical density. For example, 24, if you see this figure, that here that the linking number is applied to a closed circular DNA molecule. So then you can see that 200 base pair circular DNA here. So when it is in the relaxed form, it has 200 linking number. That means the first strand is crossing the second strand by 200 times, right? But when the strand will be broken, then this is a discontinuous deformation. So the linking number is now undefined, right? But if you will reduce the two number of turns, then if the delta LK is minus 2, then the final linking number will be 198, right? 200 minus 2. So the superhelical density of the sigma is will be delta LK is 2 divided by 200, right? Which is 0.01 for this case. Right, now we will see what is positive and negative supercoiling. So suppose this is a relaxed DNA which has a linking number of 200, means the first strand is crossing the second strand 200 times, right? Then if you reduce two turns, right, then the delta LK will be minus 2. So the final linking number will be 200 minus 2, which is 198. So as the as the linking number has been reduced, so that's why it is called. So that's why it will induce the negative supercoiling, right? And if you introduce two more turns here, right? If you will increase the linking number by 2, then the final linking number will be 200 plus 2, which is 202. That is positive. That will induce the positive supercoiling. So if you decrease the leaking number, it will introduce the negative supercoiling. If you will increase the leaking number, then it will induce the positive supercoiling. Okay, so you can tell me if you have any doubt. So next we will uh, saw that what is negative and positive supercoiling. So again, if you want to revisit that, the suppose your relaxed DNA has linking number of 200, right? Then if you will decrease the linking number by 2, then it will introduce the negative supercoiling. So the final linking number will be 198. And if you will increase 2 linking number here, then it will induce the positive supercoiling. So the final linking number will be 202, right? So this is the negative and positive supercoiling, right? And this linking number is summation of two topological property, right? Which, so Lk equals to Tw plus Wr. So Tw is the twist and Wr is the Rithe number, right? So this twist and this Rithe, that is actually calculating the degree of supercoiling, right? That's why linking number is a measurement of a supercoiling. So for example, here this is a strained element, which is a, suppose this is a relaxed DNA. Then for the large Rithe, the if you will make its, you know, the, if you will enlarge the Rithe, then it will introduce a small change in the twist, right? But if you will decrease the Rithe to 0, then for 0 Rithe, the large change in the twist will be happening, right? So that is a Riemann model for illustrating the twist and Rithe number, right? So there is an enzyme called topoisomerase which actually catalyzes the changes in the linking number for the DNA. So suppose this is your relaxed DNA, right? And then if you will remove some terms, then it will be underbound DNA. So it will introduce the negative supercoiling, right? Which will actually produce the cruciform DNA here, right? So now, if you will see the topoisomerase activity here, then if you will run the agarose gel of 3 samples, one, right? So, so in this experiment, so first panel is showing the relaxed DNA and highly supercoiled DNA. So because of this highly supercoiling, this supercoiled DNA can migrate faster than the relaxed DNA, right? Because it will take less, right? So because it can migrate faster, right? So it can, so that because it has the less hydrodynamic radius, so it can easily bypass the, you know, the agarose pore size, and it can migrate faster, right? And in the second panel you can see that the supercoiled density, so this is the direction of decreasing linking number, right? So here you can see that as we go down, the linking number has been decreased by the, when you will use the topoisomerase. Right. So how this topoisomerase work? This topoisomerase will, uh, when the DNA bind, right? So this topoisomerase has a phosphotyrosine. So DNA binds with this phosphotyrosine here. That is the enzyme DNA complex. Then this, right? So after DNA binds, an active site tyrosine attacks a phosphodiester bond on the DNA strand. So this bond will be attacked, right? So that it will be cleaved and creating a covalent 5' phosphate tyrosine DNA linkage. That is your, after cleavage you will get this, and it will use this gap to pass the second strand, right here. So that will actually create a change in the linking number, right? So after this cleavage, the second strand will be passed throughout this gap, right? So that is, so that will actually change in your linking number. right? So then we will actually start the DNA metabolism, which is actually replication. We will start. So first replication, how was it studied? That and they saw that DNA replication is semi-conservative, right? That means the if this is the parental DNA, then after one round of replication, there will be two molecules of DNA, right? Each of will have one parental strand and one nascent strand, right? So that's why it is semi-conservative. So how did they find that? So first they took the original parent DNA molecule, right, which is having this, right. So when the DNA extracted and centrifuged to equilibrium in CaCl density gradient, because those cells were grown in the media which contains the 15 nitrogen isotope, right. So that's why you will get only one kind of DNA, right, which is 15N, which is heavy DNA. And then after one round of replication, right, uh, for this replication they were incubated in media contain the 14N, right. So after this replication, it will result into two molecules which has 15N as well as 14N here also, right. So you will get only the hybrid DNA which is 15N 14N. And then after one more round of replication, which is in the second generation, then you will see that this um, so it will cause actually, so this blue one which is 15N, that will be used as a template, right, and it will take the 14N as the daughter strand, right. This 14N will combine with another 14N, this 14N will combine with another 14N, and this 15N will combine with another 14N, right, because for the replication the cells were grown in 14N medium. So now you got two 14N 14N and two 15N 14N hybrid, right. So you got one hybrid DNA which is 1514 and another one is 14 DNA, right, which is this one, right. So this experiment actually proved that the DNA application is semi-conservative. This is called as the Michelson-Stall experiment, right. So till now, if you have any doubt, you can ask me. Okay, so now we have seen that DNA replication is a semi-conservative replication, and if you see that the DNA, how DNA replication actually starts, right? So it is a bidirectional DNA replication. So here in this electron microscope, you can see that when a which is showing the replication of a circular DNA, right? So here this portion has been melted, so here this fork is fired, and the replication is proceeding in two direction. One is this direction, another is in this direction, right? So and then at the end of the replication, it will create this kind of twisting thing, right? So when the topoisomerase 4 will come, and it will release it into two DNA, right? So that's why it is called the bidirectional replication, because this fork will move in both of the direction, this and this, front and back, right? And this is example of a unidirectional replication where fork will move in only one direction. If you can see this diagram, that this red one is the newly incorporated nascent DNTPs. So they are actually being incorporated in both direction, here also and here also, right? So this is bidirectional DNA replication, right? So here basically the when the helicase will come and helicase mate the DNA, then it will then this is the direction of the movement of the replication fork, right? So this will go to melt this or denature this hydrogen bond. So DNA will be so DNA will be gradually denatured, right, which is called as the movement of the replication fork, right? And this DNA is synthesized by an enzyme called DNA polymerase, right? So which will incorporate the DNTPs here. So basically, what is happening here, that this DNA polymerase has aspartate in the active site, and it also need Mg2 plus ion for the cofactor, right? So it will, so that this Mg2 plus ion and this DNA, it has a phosphate group, right? This nucleotides, they have phosphate group. So this aspartate, it also has phosphate, the negative charge, and this phosphate group will also have the negative charge. So just to so that just to reduce the repulsion, this Mg2 plus, which is a positive charge, it will come here to act as a mediator, so that DNA polymerase can interact with the negative charged DNA strand, right, or the DNTPs, right, through this mediator Mg2 plus, right? So that's how this DNA polymerase will bind to the substrate, and it will incorporate one DNTP here, right? So this is the incoming, right? So this is the incoming DNTP, right? So that will be incorporated by the, so it will make this phosphodiester bond with the previous DNTP here, right? And then they will make a phosphodiester bond, right? So that's how DNA polymerase can add one by one the DNTPs, right? And then DNA replication is very accurate because the DNA polymerase has the two feeding activity, right? It has a 3' to 5' exonuclease activity, right? So if there is any wrong base is coming, for example, that in the template strand there is adenine, so suppose by mistake it has incorporated the cytosine here, then which will create a mismatch, right? So then this, because of this, right? So this C cytosine has a rare automatic form of cytosine C star that can pair with A, right? So if it is incorporated in the drawing strand, but this CA is a mismatch, right? T has to come here, thymine has to come here. This CA is a mismatch. So if the polymerase will go on, then it will create a mutation here, right, in the next round of replication, right, because of this mismatch. So it has to fix it. So for that, if there is anything, I mean, anything else will come except the thymine, right? Then, right, then it will not be fitted in this proofreading domain, right? So now that's how the DNA polymerase can sense that a wrong nucleotide has been converted here. So DNA polymerase at this stage it will show its 3' to 5' exonuclease activity, right? So that it will chew this DNTP, right? It will chew this nucleotide, right? And it will against so then it will create a 3' hydroxyl group here, and then it will again bring the proper, the correct nucleotide here by the 5' to 3' polymerase activity, right? So first DNA polymerase will slide back, and then again it will show its the polymerase activity to add the incorporate the correct nucleotide, right? So that is done by the DNA polymerase 1, right? DNA, yeah. So this is the error. This is the proofreading activity of the DNA polymerase, right? And if you see the bacterial DNA polymerase, it has three DNA polymerase: Pol A, Pol B, and Pol C. And if you see the subunit of the DNA polymerase of E. coli, it has the alpha subunit, epsilon, theta, tau, and some other, right? These are the main subunit of the DNA polymerase. Alpha has the polymerization activity, epsilon has a 3' proofreading activity, theta, right, and this tau has the stable template binding activity, right, and this gamma, delta, and delta dash, they are the clamp loader, right, and right, so and this xi, it will interact with the single strand binding protein, so that whenever there will be single strand DNA, the single strand binding protein will come and bind. Okay, so next we will see the nick translation by DNA polymerase 1. So when there is a nick here, for example, in the lagging strand there are Okazaki fragments, right? So between two Okazaki fragments there are gap, right? A nick. So to repair this nick, DNA polymerase 1 will bind, right? And then it will show its 5' to 3' exonuclease activity, right? So it will remove the RNA or this DNA element which is green color, right? And it will show its 5' to 3' polymerase activity to incorporate the DNTPs, which is pink color, right? And then it will extend it till here, right? After that it will be dissociated, and then this nick will be sealed by the DNA ligase, right? So that is called nick translation by DNA polymerase 1, right? And this is the DNA polymerase 3, which is the replication enzyme in the bacteria prokaryote, right? So it so this is the code subunit alpha, epsilon and theta, which has the role in binding the template and in the polymerization activity. It is associated with the DNAB helicase. Helicase will migrate and it will continuing to break the hydrogen bonds, right? So that the DNA will be denatured. So that's how the application fork will be proceeded, and this each of the code subunits will with associated with the beta clamp, right? So that it can hold the template DNA here and it can incorporate the incoming nucleotides, right? So this is a PDB structure of a crystallography structure of a DNA polymerase which is bound with the DNA, right? So DNA replication So the DNA replication with it will start in a specific replication origin sequence which is called ODC for bacteria or prokaryote. So it is composed of tandem array of 313 base pair sequence, right? This is a binding site for the DNA A and 4, right? So these 4 green blue colored regions are the binding sites for the DNA A, and it contains the consensus sequence which is TT80CCACA, right? So how this application has been started? So for E. coli, it has a supercoiled template, supercoiled plasmid. So suppose this region is the OEC region. So it has a 313 base pair repeats, this one yellow color, right? And in the downstream there are 4 9 base pair sequences, right? These are the 4 9 base pair sequences, which is the binding site for the DNA A. So first DNA A will come along with the ATP and it will bind to this 9 base pair sequences, right? So DNA A will bind and it will make a loop, right? Then HU will come. HU is the histone-like protein. So yeah, so HU will come along with the ATP, and then the DNA B and DNA C will come. DNA B is the helicase and DNA C is the loader. So this DNA C will load the DNA B helicase, right, here at this to H, right, and then DNA B or the DNA B helicase, it will start denaturing the DNA so that the fork will be melted, right, and now the replication has been initiated, we can see, right. So this is the replication fork movement, right. So suppose this is your suppose this is your parental DNA, then this DNA topoisomerase 2 or DNA guidance will first come, it will bind here, then as we saw that DNA helicase will be recruited and then it will start melting this fork, right, so that the replication fork will start in this direction to move, right, so it will denature the hydrogen bonds and it it will move towards this direction. Right, that is the direction of the replication fork movement, right? And then DNA helicase will start going in this direction to denatured this hydrogen bond so that the replication fork can move, right? Now in the leading strand, this is the 5.3 prime, so the in the direction of the replication fork movement, the direction is 5.2 prime, so it can easily synthesize the leading nascent strand, right, by DNA polymerase 3. But for the lagging strand, the parent end is 3 prime to 5 prime, and the right and the in the direction of the replication fork movement, the direction is the 3 prime to 5 prime, right? But DNA polymerase can only elongate in this in the direction of the 5 prime to 3 prime, right? It cannot do from 3 prime to 5 prime. So for that, there will be a RNA. So this DNA will be looped, right? And there will be a RNA primer. So then it will, so that the after the looping around, the direction will be 5 prime to 3 prime from this RNA primer, and then it will make this lagging strand, right? So then again the polymerase will collapse, and then the next portion of the DNA will be looped, right? Again this second Okazaki fragment will be formed, right? So that is called lagging strand synthesis by the same enzyme DNA polymerase 3, right? So that's how the DNA elongation in the DNA replication takes place. So this is the image where you can see the DNA synthesis of the leading and lagging strand. After origin firing, you can see that replication fork is moving in this direction, right, from this to this, right to left, right. In this direction, DNA helicase is denaturing the hydrogen bonds here, right. So now the leading strand, if this is so if this is 3 prime to, so yeah, so if this is 5 prime to 3 prime, then the nascent strand will be 5 prime to 3 prime in this direction, right? So that can be easily synthesized by the DNA polymerase because the direction is the 5 prime to 3 prime. But for the opposite strand, this is 3 prime to 5 prime. So for nascent strand, it will be 3 prime to 5 prime in this direction of the replication fork movement. So it cannot do the replication. So for that, this parental strand will be looped, right? So after this looping, there will be a RNA primer will be there, and now this direction is the 5 prime to 3 prime, right? So that's why in this direction, the replication can be possible. It will incorporate the nucleotide, right? And after some times, it will collapse, right? And then again, it will, for example, here, yeah, yeah. So after collapsing, the new primase will come. New primase will make the new RNA primer for the next Okazaki fragment, and then again it will incorporate DNTPs in this direction here, right? And it will release, right? So now the new beta clamp will be loaded onto the new template primer, right? And then the old beta clamp will be discarded. So this loop will be released, right? So this is the first Okazaki fragment, and this is the second Okazaki fragment, right? So that's how the replication occurs in both leading and lagging strand, right? Now the final step in this synthesis of the lagging strand, because as we saw that in the lagging strand there will be multiple Okazaki fragments here. There will be multiple Okazaki fragments, right? So now they will have a gap between them, which is nick, right? So in the lagging strand, this is the link between the two consecutive Okazaki fragments. So now to, as we talked about the nick translation, so the same process will be applied to fill this gap, right? So here DNA polymerase 1 will come, it will uh, show its 5' to 3' exonuclease activity to remove this RNA chunk, right? And it will replace it with the DNA, right? DNTP. So it will remove the ribosome, ribose RNPs, RNP, which are the portion of the RNA, and it will incorporate the DNTP by using its 5' to 3' polymerase activity, right? And then it will extend it. So after extending from from here, it will be collapsed. So now there will be a nick. So nick will be translated from here to here. So now this nick here will be sealed by the DNA ligase, which will use ATP as a cofactor, right, to seal the nick, or it can also use the NAD plus as a cofactor. For that case, enamine will be the byproduct, right? So this is the mechanism for the DNA ligase action or the DNA ligase activity, right? So till now, if you have any doubt, you can ask me. Then we will discuss the mechanism of the DNA ligase. So after that, we will see that how the mechanism of the DNA ligase action, how it can seal the gap. So this is a nicked DNA, right? One strand is there is a phosphodiester bond breakage, right? So now this enzyme, which is DNA ligase, that will use NAD plus or ATP as a cofactor, right? So it will remove the PPI from ATP, right? So that it will conjugate the AMP form. Or for if we so if it is using NAD plus, then it will remove the NMN, right? From that, right? And then it will use the NMP. So now for the ATP, if you will see, there will be a enzyme AMP complex, right? So first of all, what will happen that uh, this AMP will be transferred to this nicked DNA along with the phosphodiester bond. So yeah, after that, this phosphodiester bond, right? So this nicked, so this enzyme AMP, it has this AMP along with this enzyme. So this PO minus, that will actually act as a nucleophile, right? So this OH, it has a 3' OH of the previous nucleotide. It has a non-parallel electron, so it will make a nucleophilic attack in this electron deficient phosphorus, right? So that it will be a sealed DNA, right? So this nick is sealed. It will make a bond, and this bond will be broken, right? So now the conjugation between this AMP and the phosphate group will be broken. Now this phosphate group will be make a phosphodiester bond with the previous nucleotide OH group, right? So it will release AMP here, right? So now this DNA gap is sealed. That's how the DNA ligase enzyme is using AMP, which is coming from ATP, right, as a cofactor to seal the gap here, which is called the nick, which is sealing the nick, right? So how the DNA replication gets to be terminated? Then so the process is yeah. So suppose this is two opposite direction fork, counterclockwise fork and the clockwise fork. They are moving into the opposite uh, direction, and then when they come here, then this is actually indication of the completion of the replication here, right? If you will zoom out this. So now that this is called the catenated chromosome. So now DNA autopoisomerase 4 will come and it will just uh, release these two chromosomes, right? So this is called the separated chromosome. So now it has been produced from this circular DNA, right? From when the two replication forks will come here, then DNA topoisomerase 4 will produce the two separate chromosomes. For example, two separate here, it will produce the two circular plasmids, right? So that is called the replication termination, right? So this is, and for the prokaryotes, suppose this is your ODC and there is one clockwise fork, one is anti-clockwise fork. So these are the, when they will meet here, TAR B, TAR D, TAR A, these are the counterclockwise fork trap. So this fork will be trapped here, and TAR C, B, F and G, these termination sites will be the trapping site for the clockwise fork, right? So when they will meet here and they will be trapped, then the DNA topoisomerase 4 will come and it will decatenated the chromosome so that they will produce the separated chromosome right here. So two circular plasmids will be produced. So that's how the DNA did. So that's how the DNA replication will be terminated, right? So now we will see a summary of the DNA replication, right, which is chromosome partition in the bacteria which we studied. That this is the bacterial chromosome, right, which is circular plasmid. So this is origin of replication. So from the origin of replication, replication fork will start moving to opposite sides. One is at clockwise, another one is at the anti-clockwise here, bidirectional replication, and then the cell will start elongating and replication continues, right? And after that, when they will reach into the termination site, then two forks will be trapped and they will be separated. Chromosomes will be separated to produce two chromosomes or two plasmidia, and then the cells will be divided, right? And then each of the cell after division will have one chromosome or circular plasmid. That is called chromosomal partitioning in the bacteria. Right, so that was for the DNA replication, and for the DNA replication in the eukaryote, that is more complex. There are, so for example, in bacteria the origin of replication is called as ODC, but for eukaryote it is called as autonomously replicating sequence, or ARS, or replicators, right? And they have been studied in the East. And so initiation of this replication in the eukaryotes require a multi-subunit protein which is called origin recognition complex ORC, that can recognize the origin or the R site, right? They can bind to the R site, and then after interaction of the ORC and the R site, several other proteins will come, right? Which are called the cell division cycle proteins or CDC proteins, right? They will bind to the ORC and they will mediate the platform for loading of heterohexamer, right, which is called as the mini chromosome maintenance proteins, right, or MCM, right, MCM2 to MCM7, which is a 2, 3, 4, 5, 6, 7. There is, which is a 6 subunit uh, hexamer that will be loaded, and which will actually, as it is a helicase, so it will start melting the DNA, right? It is just equivalent to the DNA B of bacteria, right? So it will start denaturing the DNA, right? And then it will start the elongation of the replication, right? So that is the difference in between the replication initiation in the eukaryote and the prokaryote, right? So that's all for the replication we have studied. So if you have any question, you can ask me. Mayank, do you have any question regarding to this topic?
No, sir. But I have a one query.
Yes,
Are
yes.
you recorded this particular lecture because I am pretty late when I joined this
Yeah, yeah,
lecture?
yeah. Actually, all the lectures have been recorded, so that will be uploaded very shortly, very soon.
Because I want
Yes, yes. Tell me.
to see the first half of the first half of the lecture also for any query
Ha
to
ha ha
understand
ha.
anything.
Lectures have already, all the lectures have already been recorded, right? So it will be uploaded soon, and then you will see the from the very first lecture, right? So
Thank you, sir, for your time and class.
and any doubt for from today's session you had, you can ask me.
Okay. I will mail you if I have any
Okay, okay,
doubt.
fine, fine, sure, sure,
Thank you, sir.
sure.
Good
Okay,
evening, sir.
okay, okay. So in the absence of any other further question, I think we can close the session. And Mayank, the next session will be, uh, although I will send all of you the mail, but still the next,
Okay, sir.
but the next session will be on the next Friday from 8 to 10
Okay,
p.m.
sir.
Right?
Okay, sir.
Yeah, in the same meeting link.
Okay, sir.
Yeah, yeah. Okay, okay. So do join in the next session, and so we can end the session here. And okay, so see you on the next week. Bye.
Thank you, sir. Good evening.